Hello. Hello. Good morning everyone and a very warm welcome to day 4 of the national level 5-day faculty development program on artificial intelligence and emerging trends in healthcare and agricultural business segment, organized by the School of Science and Technology, the Neotia University. Over the past 3 days, we have explored an exciting array of topics from biomechatronics and AI in clinical diagnostics to robotics, agribusiness and computer vision. Today promises yet another set of engaging and intellectually stimulating session with expert talks focusing on deep learning in bioinformatics and healthcare and predictive analytics in medicine. So let us now begin session 1. Speaker of the day is Dr. Ranojit Sarkar. Dr. Ranojit Sarkar is an accomplished academician and researcher with nearly 20 years of experience in teaching and research. Dr. Sarkar completed his MSc in bioinformatics from the University of Pune in 2004. His thesis work was titled Bioremediation of Low-Density Polyethylene, LDPE. He has authored over 15 publications and in international journals and delivered talks at numerous conferences. Dr. Sarkar is a recipient of multiple prestigious scholarships, including the ICCR, Government of India and the G.N. Ramachandran Fellowship, which underscores his contribution to scientific research and academic excellence. Please join me in welcoming Dr. Ranojit Sarkar. And on this note, I request our Dean, Professor Partha Mukherjee, to felicitate Dr. Sarkar. Today, Dr. Sarkar will be speaking on a fascinating and timely topic, deep learning for protein structure prediction, AlphaFold, Rosetta Fold. Over to you, Dr. Sarkar.
Thank you, ma'am. Am I audible? Am I audible? Good morning, dear sir, and my dear colleagues. So, this session, actually, I am looking it as an opportunity to share or discuss some points among us. And my sole purpose is looking forward, we can work together across the department, like agriculture, pharmacy, and other allied departments. So today, we will discuss some key points in biotechnological research, and that is in silico mode. So today actually we are interested in proteins. Now I will talk a bit why the proteins are so significant, so important in our research. So before that, first let us revisit the proteins. So what are proteins? You know that proteins are one of the essential biomolecules. So you know that biomolecules are either branched or unbranched. So like DNA and protein, they are unbranched biomolecules, whereas carbohydrates and fats, they are branched type of biomolecules. Now, why protein is having so much focus over the other biomolecules? Because it performs diverse functions, you can see, as enzymes, signaling transports, and immune defense. And you know that almost all the enzymes are proteins, except some ribozymes or DNA enzymes. They are very few, but almost all the proteins are structurally, enzymes are structurally protein molecules. And you also know that without an enzyme within the biological system, no function can be carried out. Right? So now, this function of this protein are actually determined by its 3D structure, not just the amino acid sequences. So let me tell you that it is a polymer. As I said that it is a biopolymer. These biomolecules are biopolymer. That means they have some repeating units. So in case of protein, you know there are 20 repeating units. So these are called amino acids. Though there are hundreds of amino acids in the cells, but out of these hundreds of different types of amino acids, only 20 has been selected for protein synthesis. Now till date, we do not have any idea that why nature has selected these 20 amino acids only. We have yet to find that answer. Okay. So now let's see that why structure matters. So structure determines how a protein works. We know that the journey of the protein folding, you might know that it's a complex process. Until today, we do not have an idea that how actually a protein folds into its three-dimensional structure. Why I am sharing all this basic information with all of you? Because we belong to diverse departments, diverse domains. So if we get the inside information, then from our expertization point of view, we can think about some problems and we can work together. That's the objective. So that's what I was talking about, that the protein folding is a serious problem that is still mysterious to us. And if we can solve the protein folding problem, you can assume that almost 70–80% disease can be solved, like prion, Alzheimer. So these are the diseases that is related to protein folding problems. So the journey from after the translation, the linear structure of amino acids, the polymer, we are talking about the polypeptide chains, that actually subjected to a complex, I repeat the word complex, three-dimensional structure. And a single million types of complex three-dimensional structure is possible. But out of that, only one is functional. That is termed as native structure state. Now the question is that, how come the nature know that which one is the native structure? It cannot try it out. If you see, if you changes the angles, like say torsion angles, if you see that if there is the wall and floor in between, you can see that there is 90° torsion angle, right? So here this is the floor and this is the wall. The torsion angle, the dihedral angle is 90. Now if you convert it to 45, the shape of this room will be changed. So that's why Sir Leventhal, he calculated if a protein is composed of only 100 amino acids and 10 conformation per amino acids only, almost, of course there is n number of conformation possible, but only 10 he calculated, and he calculated that a protein will take 10 to the power 65 years to fold into its correct structure. But that is not possible. We know that protein folds within femtosecond in the system, right? So that means nature knows the pathway. We don't know. That means we have to find out what are the exact pathways. So you can see that even a single mutation affecting the structure and cause disease. Like you know, in case of sickle cell anemia, in that case the protein is almost 156 amino acid length, okay, and only one mutation is enough to cause the sickle cell structure. So drugs binding sites and enzyme active sites are structurally defined. So where the drug will bind with a protein, so if you want to block the function of a protein, that means your drug molecule has to go and bind at a particular site of the protein. And that site is very, very specific. If there is any changes, the drug will not be able to bind. Similarly, for the enzymes, the substrate has to bind at a particular site, and the structural geometry plays the key role in enzyme substrate binding. So that's why understanding the structure is the prime focus in molecular research. So now we will focus on the experimental methods and of course its limitations. Okay, so we can solve the structure. You can ask that if you have the protein, you can solve its structure, but through experimental methods, you are right, we can solve it. We can solve by X-ray crystallography, NMR, and cryo-EM are the gold standards. But there is some problem. What are the problems? See, they are very, very expensive. There are only few labs over the world. I am not talking about only India. Over the world, they can carry out X-ray crystallography and NMR. Okay, so few limited labs are there. So they are very expensive. They are also time consuming. So to solve a protein structure through X-ray crystallography will take around 5 to 6 months. Now, it's not feasible for all proteins also, like the transmembrane proteins. We cannot solve the structure of transmembrane proteins by X-ray crystallography or NMR. So these are the huge limitations in case of the solving of protein structure. So we have discussed that why the protein structure matters. Okay, so now how we can go about this critical situation? So now let's see the structural knowledge gap. So what is the structural knowledge gap now? You see that there are tens of millions of protein sequences are known. At this day, we know tens of millions of protein sequences, and we give thanks to the genome sequencing project. So you know that we started with the human genome sequencing in mid-90s. Around 1985, we started with the human genome sequencing, and we had the target that by 2020, we will complete the sequencing of human genome. We have 46 chromosomes, and the 46 chromosomes are distributed over 13 labs over the world. In 13 labs, the genomes are distributed, and the target of finishing of the genome sequencing was 2020. But you will be astonished to know that we completed the human genome by 2003. Okay, so before time, almost 17 years before we completed the sequencing of human genome. So what happened in between? In between, because of the advancement in technology, especially in molecular biology technology, we could complete this immense task before the time period. And we thought that when we will get the genome sequences, our job is done. So because we know that the phenotype, all the things is connected to the genotype. So if we have the genome, our job is over. we need not to go for further research. We can relax. But unfortunately, we could know that this was just the beginning. After getting the human genome. Why? After getting the human genome, it was the beginning of the research because we have got 3 billion base pairs. You know that genome is composed of 4 bases. It's a polymer and it's composed of 4 bases only. A, T, G and C. Adenine, guanine, thymine and cytosine. So it's common for all the organisms. So we vary. The living organisms vary because of only the patterns, not the composition. So any living system on this earth, they have the same genetic material composed of these 4 units only. That is A, T, G and C. But how we are varying? We are varying from one to other. Okay, so this variation is due to the difference in patterns only. That means nature has encoded the pattern and our job is to decode that. So that is the opportunity with us that we can work on. Now you see that these 3 billion genes, 80 GC bases we have and we cannot use that. Why? If we write down the 80 GC, these 3 billions, you have seen some of you that biology students are moving around with the books of volumes like this, Leninger. So this is the volume of Leninger. Chandra might know that. Okay, so if we write the 3 billion base pairs, it will be 20 equal volumes of Leninger. And it is written only 80 GC. And it is of no use. It is of no use. Why it is of no use? Because what is the use? Now we have to find out what are the functional units. We know that only 2% of our genome is functional. Only 2%. 98% is not directly functional. So these days we do not call them non-functional unit, but they are actually helping in organization of the genome. Not directly they are coding protein. So protein is coded by only 2% of this 3 billion base pairs. Now how we can identify those? So because of advancement in silico techniques, computational techniques, in computational biology, we could find out the genes and proteins in human genome. So now we have 30,000 of genes and around 1 lakh of protein in humans. So all this data we have to organize in database form. So that is the different domain of databases. So today we will not focus on databases. Okay? So in another day we will take an opportunity to discuss among us regarding databases. So only you see that out of these millions of proteins, only fractions have known 3D structures. Only fractions. Now we said that the 3D structures are the key for any research in drug discovery and drug development, or understanding the mechanism of the disease. Okay? But on the other hand, we have only fractional proteins of the known protein that have known 3D structures. So what we need the computational methods to fill this gap. Now let us see, before going to the methods, briefly that what are protein structures and how we actually represent them. So now you can see that these are the amino acids. So you can see that these are the amino acids. Okay, see there are 2 amino acids. The amino acids basically they have a common structure. You see the common structure among the amino acids, only you can see that R is written there. That means this is the biochemical group that is varying from one amino acid to structure is common. Thanks to God, we need not to remember a complex structure. Only we can remember the R group and we can remember the structure of amino acids. So you may see that two amino acids are fused to form a dipeptide. Here you can see that there is dipeptide structure and a peculiar bond has been formed. That is the CN bond you can see. So this is not the usual CN bond, it's not the amide bond because it has a partial double bond characteristics. It's neither a single bond nor a double bond. Now you can ask me that why I am discussing some chemistry. Okay, because it's very important to understand this chemistry, to understand the structure of protein. Now you see that because of this partial double bond characteristics, okay, the CO and NH, they are coplanar. That means they cannot move. No rotation is possible in between CO and NH. Okay, so this dihedral angle is termed as omega angle. That means omega is 180. But around the omega bond, the rotation is possible. That's why we get diverse protein structure. So now this, again, number of amino acids can be added in this way and you can get the polymer, and this polymer you can see below, we can represent this polymer as single letter codes. Here M actually stands for methionine, G for glycine, L for lysine accordingly. So each of these 20 amino acids, they have a single letter code as well as 3-letter code. But in computational study, most of the cases, we use a single letter code. Okay, so this is the primary structure of protein. Now, let us understand the structural complexity so that we can apply our computational techniques to understand its 3D structure. You see the level of structure. First, you can see a ribbon-like structure. So this is the actually polymer we are talking about. So number of amino acids will join one to the next one and they will form a long unbranched chain. Okay, long unbranched chains of amino acids. So it will have the direction. The two ends are not equal. One end will have a free amine group and the other end will have a free carboxyl group. So we call them N-terminus and C-terminus accordingly. Now, ultimately, the 3D structure is functional, as I say. This 3D structure is functional. Now, in between the primary structure and tertiary structure, it will form some local folding. And this folding is very regular. Very regular. So almost all the proteins we have found so far only 2 types of local folding. And that is one helix. You can see on the left side, this is the helix type of structure. In helix, there will be a hydrogen bonding pattern within the chain. We call it intra-chain hydrogen bonding. And its pattern will be I equal to I plus 4. That means the first amino acid will form the hydrogen bond with the 5th amino acids. And this pattern will go on and it will form the maximum number of hydrogen bonds so that the structure can be stabilized. So we find the single-stranded alpha helix and a beta-pleated sheet. Beta-pleated sheet means the interchain hydrogen bond is formed and number of strands, they are stabilized by forming hydrogen bond among each other. So that is the beta-pleated sheet. Now the critical point is that how this alpha helix and beta-pleated sheet are arranged topologically, geometrically to get the 3D structure. As we said say that Sir Levinthal calculated the time frame, and that is termed as Levinthal paradox. Okay, so that is the most critical point, that how system actually understood that this particular form of 3D structure is need to be formed. The another structural complexity, you can see that in the bottom, the coordinate structure. So if your protein is having more than one chain, then only coordinate structure will be functional. Otherwise, it's the 3D structure. So if the protein is having more than one chain, then tertiary structure is not functional. It's the coordinate structure that will be functional. So again, it is a complex level where we may go wrong while predicting the protein structure. So that is the critical point. So now we have revisited the structural complexity level. So this structural complexity level actually comes from this understanding of this geometry. That's what actually we need to understand a bit of this geometry to understand that how we can calculate the structure, 3D structure of a protein.